Hi, I'm Viktoria Johansson, and I'm going to talk about cerebrospinal fluid markers in twins with schizophrenia and bipolar disorder. I'm a postdoc at Karolinska Institute in Stockholm, Sweden, and I also work as a clinician right now with patients with ADHD. I have no conflicts of interest to declare. First, I'm going to tell you about STAR, the Swedish Twins and Relatives Study. It's a twin cohort with twins with schizophrenia and bipolar disorder, and it was established by Christina Hultman at Karolinska and Tyrone Cannon, now at Yale University. The twins were recruited from the Swedish Twin Register. This register was established already in the 1960s. And all twins that were born in Sweden from 1886 and forward are in this register. And the twins were recruited nationwide in the whole Sweden, from the whole Sweden. And in this study, in total, 462 twins participated. And they went through clinical assessment. They filled out questionnaires on personality traits, and symptoms were assessed, for instance, negative and psychotic symptoms. They also went through neuropsychological testing, brain imaging with fMRI, birth data was collected, and blood samples. And from this larger cohort, we recruited twins for a smaller study on cerebrospinal fluid. We focused on recruiting discordant twin pairs, where one twin has either schizophrenia or bipolar disorder, and the other twin is not affected. So we recruited 25 complete pairs and one single twin. And we ended up with some concordant pairs and not affected pairs as well. So why would you like to study twins in the research? As you know, monozygotic twins are identical with 100% identical genetic setups, while dizygotic twins are as equal as siblings. And they share environment, or more, early environment. And differences in the monozygotic twins are assumed to be caused by the unique environment, so that's why they're very interesting to investigate. So in this study, the twins went through lumbar puncture, so the twin pair came together to St. Göran Hospital in Stockholm to undergo lumbar puncture. And the first drops were collected for a microscopy examination with scanning electron microscopy and put on a filter for gold coating. And we also collected CSF for later biomarker analysis. So this picture is just to show you how a microscopic picture can look like. We have several projects on the CSF twins. We have analyzed microscopic structures in cerebrospinal fluid. We have also analyzed microparticles and microglia and neurodegenerative markers, and quinine metabolites, cytokines, and also a project on proteomics under analysis. And now I'm going to talk about the project on microglia and neurodegenerative CSF markers. In this analysis, 35 twins were analyzed. And we analyzed the markers CD14, YKL4, and MCP1, and also amyloid peptides, amyloid precursor proteins, and tau proteins. And our objective was to analyze the heritability of all those markers and to perform proven co-twin analysis in the discordant twin pairs, and also to relate the CSF markers to psychotic symptoms and other personality traits. And this is some descriptive data on the sample. I divided them into monozygotic and dizygotic twins. You can see the mean age is quite high, 55. Yeah, and the schizophrenia bipolar disorders the diagnoses are quite evenly distributed between monozygotic and dizygotic pairs. So in the heritability analysis, we performed an ACD analysis where you can estimate the proportion of variance of a trait that is due to additive genetics, common environment, unique environment, or non-additive genetics or dominance. And as you can see here, on the left-hand side are markers such as amyloid precursor proteins, the tau proteins, and also the CSF serum albumin ratio. And in those markers, there are a higher degree of genetic influence. Those markers, are common in Alzheimer's research. While the markers related to inflammation, such as CD14 and MCP1 and so forth, they are more influenced by, or less influenced by genetics, as you can see here. And then we continued with analysis on the discordant twin pairs. And the only marker that showed significant results in this analysis was CD14. And as CD14 is released by macrophages, and it's a co-receptor that detects bacterial LPS. So it's related to immune functions. Previously, higher levels of SCD14 was found in plasma in patients with schizophrenia and also bipolar disorder. And we also found higher levels of SCD14 in the probands that had either schizophrenia and bipolar disorder in relation to their not affected co-twins. And this difference was statistically significant. So we separate the analysis into the dizygotic twin pairs and the monozygotic twin pairs. But then we found that the association was only significant in the dizygotic twin pairs. And this finding indicates that the relation between SCD14 and the disease is affected by genetic confounding. It was not good. And then we continued to look at the differences within the twin pairs of SCD14 in relation to differences within the twin pairs of the different psychotic symptoms and personality traits. And here we found that an association between SCD14 and negative psychotic symptoms, schizotypal personality traits, and impulsive traits. But also here, the association was only significant in the dizygotic twins, and they completely disappeared here in the monozygotic twins. And this indicates that this relationship was also affected by genetics. So to conclude here, our findings here strengthen previous findings that there is an association between SCD14 and schizophrenia and bipolar disorder, which is in line with the hypothesis of immune activation in those disorders. We also found, this association between SCD14 and psychotic symptoms, and a novel association also to impulse in personality traits. But these associations, or our results show that genetic mechanisms are involved in those associations, both in schizophrenia and bipolar disorder. So these results may be in line with the hypothesis that, for example, schizophrenia, but also bipolar disorder, are multi-system disorders. So higher SCD14 and higher inflammation is one symptom in in this multi-symptom disorder. So the limitations are that this is a very small twin sample because it's very hard to find, especially discordant pairs are very rare. And also this heritability analysis, that it's a problem when you don't have, when you have discordant pairs because they will affect the results. The ideal thing would be to have a whole cohort of healthy twins when you want to do that analysis. So the findings have to be interpreted with caution, but for the future, more twin studies are needed in research, where you can estimate if you have an association, you can find out if genes or environment are involved in that association. And also larger twin studies on CSF. Maybe you can collaborate with other twin cohorts on schizophrenia and bipolar disorder, and to increase the power. It's also possible to use sibling design to get more pairs, sort of. So finally, I want to thank. Here are some people involved. Here is Kristina Hultman from Karolinska that started the cohort together with Tyrone Cannon here with this research group at UCLA. And the twins came to St. Göran Bipolar Hospital to undergo the lumbar punctures. And here is my clinic in Stockholm, the ADHD clinic. Yeah. So thank you very much for listening. I'm really happy to be here.